Hi. So, in previous session you have learnt about the discovery of this various kind of DNA sensors and their adapter molecule, more precisely that discovery of a sting, and you have also learned that this DNA sensing is basically dependent on TBK1 and IRF3, and this signaling axis basically induces the type 1 interferons. Okay. So, in this session we will discuss about the various DNA sensors and their signaling pathways. Okay. Because it is very interesting that how various signaling pathway converge to the TBK1, and that basically induces type 1 interferon. In addition, this also activate NF kappa B to induce the pro-inflammatory cytokines. Okay. So, the DNA sensors basically there is some obvious property of DNA sensors. So, DNA sensor basically bind with DNA, and when the DNA go inside the cell, basically this perturb the DNA homeostasis. Okay. And basically this will activate the cascade of signaling pathway, okay, in order to induce innate immune response, and this innate immune response is basically the production of type 1 interferon and pro-inflammatory cytokines. Okay. They can in addition to the production of type 1 interferon and inflammatory cytokine, they can also induce the cell death. Okay. And in technical term we call it as a apoptosis. Okay. And this is if you see, the apoptosis is also very important defense mechanism. Here I will just elaborate a bit more. Why? Because generally when we see the cell apoptosis, we may think that it is not good for the host, but in case of virus infection, cell death is a very important defense mechanism. Why? Because when virus infect the cells, they hijack the cell, right, and then they use this host cell machinery in order to make their copy. So, there is no other way to stop the virus rather than using programmed cell death. Okay. If the cell trigger the programmed cell death, then the whole cell will die, and along with the virus will be virus replication or the metabolic activity which is used by which is skewed towards or skewed for the replication of virus will be stopped. Okay. So, this is the best way to stop the viral replication, to kill the virally infected cell, and in that way the progeny will be not produced more, and then eventually the viral load will be reduced in the host. Okay. So, it is something like that. You know that in order to, so our body or our host basically in order to protect the whole host, they kill some cell. Okay. So, it is something like that. It is a very old saying, probably you may are aware that in order to protect the pond, you need to take out that dead fish. So, something like that. So, similar strategy is adopted by our body. Okay. So, this signaling specificity of DNA sensors is attributed to various factors, and that includes the length of DNA molecule, length in terms of base pair, the 3-dimensional structure. So, the 3-dimensional structure is very important. Okay. And you know that mainly RNA makes a various kind of 3-dimensional complicated structure, but DNA do make some or various kind of three-dimensional structure. If you remember, the various kind of DNA are existing. Okay, although in physiological condition, B form of DNA is a most predominant structure. Okay, so in some cellular processes, maybe these structures are changing, and then this may be sensed by the DNA sensor. Okay. More precisely, the DNA from another microbial pathogen, or which includes bacteria as well as viruses, probably they have a much more diverse structure, and that can be sensed by our DNA sensors. Okay. And sequence of this foreign DNA, so length as well as sequence and three-dimensional structure, these are the key point in order to sense the DNA. Subcellular localization of a DNA molecule is also very important. For example, in general, the DNA molecule is not present in cytoplasm if you under normal circumstances. Okay. So, if the DNA is localized in cytoplasm, then it will be sensed by the DNA sensor, or it will be considered as a foreign. Okay. Another is methylation status, as I have explained you in previous session, that CpG region or CpG motif DNA, they are hypomethylated in case of or unmethylated in case of microbial pathogen and viruses. Okay. However, in case of mammalian or host cell DNA, this DNA is highly methylated, and that is a kind of distinguishing feature in order to induce the appropriate innate immune response against the foreign DNA. Association with histone and non-histone chromatin binding protein with cytotoxic DNA molecule. So, you know that this bacterial DNA or viral DNA, they are not associated with histone protein. However, in our case, in mammalian cell, this is the DNA is present in bound form with the histone. Although in bacteria some histone like protein is there, but it is not histone. Okay. And those distinguishing feature are basically sensed by the DNA sensors. So, DNA sensors could be DNase 1, which can be cleave the single stranded DNA and double stranded DNA in extracellular space. So, this is also protective response, try to understand. So, DNAs are which is present extracellular space. So, if some viral DNA will be there, or some bacterial DNA will be there, this will be cleaved by the DNase 1. Okay. This can be also cleaved by DNase 2 in extracellular space, and if the DNA is translocated in the endolysosome, so you know very well this will be sensed by the TLR9. Inside cell there are various kind of sensors, and we categorize these sensors in two major part. One is cytosolic DNA sensors. Here you can see these are the cytosolic DNA sensor, that is C-gas, IFI16, DDX41, M2, PQB-P1 and DAI. Okay. So, these are the basically a cytosolic sensor, they sense in cytosol outside the nucleus. Okay. In addition, the DNA sensors are also reported in nucleus. Okay. Or there are nuclear DNA sensor, and they are IFI16. If you see IFI16, this basically senses the DNA molecule in cytosol as well as in nucleus. Okay. And I will show you in subsequent slide one of our work that DNA, this IFI16, which is sensing DNA, can also sense influenza virus RNA. Okay, this is our work, and this is inducing the a kind of cell death which we call it as a pyroptosis. Okay. In addition to IFI 16, there are various other nuclear DNA sensors like BRCA1, RED50, MRE11, DNA-PK. Okay. So, these are the so here you can see in all situations the DNA can be sensed outside the cell, okay, in endosome, okay, in cytosol as well as in nucleus. If you see very carefully, then our cell is fully guarded against foreign DNA. Okay. And if foreign DNA will come, then it will be either digested by enzymes or it will be basically sensed, and there will be a production of inflammatory cytokine, type 1 interferon, and one more very important response will be that the cell will undergo apoptosis. This is another possibility. So, the cell or outside cell and the cell is fully guarded against the foreign DNA. Now, I will take up each sensor. The first is TLR9, and you are very well aware about the TLR9 as I have discussed in previous session when I have discussed toll-like receptors. Anyway, I will again explain. So, this TLR9 basically sense the CpG DNA, okay, inside the endolysosome, okay. And please note, it is not depicted in this image, but here there is a one important note that this DNA molecule is sensed by post translationally modified TLR9. Okay. There are some enzyme known as cathepsin family protein. They are protease. So, these protease basically cleave this native receptor TLR9, and after cleavage this protein basically sense the CpG DNA, and this is a active form of TLR9. The receptor, the whole receptor itself, the naive receptor whole is not able to sense the CpG DNA. Okay. So, before sensing it undergo cleavage by cathepsin family proteases, okay, and upon sensing you know that this will activate NF kappa B through IKK pathway, okay, IKK kinase will be activated, and then this will activate the NF kappa B to induce the pro-inflammatory cytokine. On another hand, basically upon sensing, this will be also recruit the MyD88, and this MyD88 activates IRF7. I have told you this is a transcription factor. So, MyD88 directly activate IRF7 via various protein complexes. Here you can see there is a IRAK1, IRAK4, IKK alpha. So, all these TRAF3, all these guys basically activate the IRF7, and that will induce the type 1 interferon. So, this TLR9 basically after cleavage they get dimerized, and then this will bind to the TLR9 in pH dependent manner, and then all these signaling are induced. There are various TLR9 ligands. Here you can see that there is a herpes simplex virus. There are two types of herpes simplex virus, and the DNA is sensed, the TLR9 sensed the DNA from these two viruses. Herpes papilloma virus, varicella zoster virus, Merkel cell polyma virus, cytomegalovirus, Kaposi sarcoma, associated herpes virus, Ectromelia virus, Epstein Barr virus, HIV, the DNA intermediate. So, when HIV infect, then there is a stage they make a DNA intermediate, and after making this DNA intermediate, this DNA is integrated into the genome of a host cell, and we call it as a provirus. So, that intermediate is also sensed by the TLR9. The DNA molecule from various bacteria, Neisseria meningitis, Mycobacterium tuberculosis, and fungus, that is Aspergillus fumigatus. So, the DNA from viruses, bacteria and fungus is sensed by TLR9, and that induces the type 1 interferon and inflammatory cytokine. Another very important DNA sensor which directly interact with DNA molecule is cyclic GMP AMP synthase. Okay, and this cyclic GMP and AMP synthase, we call it in short C gas. Okay, so this C gas also senses the cytoplasmic DNA. Here you can see that the C gas basically binding with DNA molecule, and this triggered the conformational change that induces the enzymatic activity. So, you can understand this is an enzyme. Okay, and this enzymatic activity converts GTP and ATP into CGAMP. Okay, and this CGAMP which is basically 2 prime 3 prime CGAMP product basically bind with the sting which is present. You remember the sting molecule which is act as a adapter, and this sting molecule is basically present on endoplasmic reticulum, and this sting interact with TBK1, and then this TBK1 or IKKI basically phosphorylate this interferon regulatory factor or IRF, particularly IRF3, and this will induce the type 1 interferon. This also activate IKK complex and subsequently activate NF-kappa B. Okay, and this NF-kappa B basically produce pro-inflammatory cytokines. Okay, and chemokines as well. So, in that way the C gas is sensing the DNA molecule. Here you can see it is quite interesting. The C gas is an enzyme, and upon binding with the DNA molecule, this molecule basically produce an intermediate, which you can see that this is a 2 prime 3 prime CGAMP. So, this intermediate basically activate the sting molecule, and indirectly this is sensed, the DNA is sensed indirectly. Okay, and it induces the innate immune response. Okay. Another is there are immune sensing of cytoplasm double stranded DNA as a non-self nucleic acid. So, here you can see that there are unstructured double stranded single stranded DNA, long double stranded DNA molecule. Okay, and there is a one molecule known as TREX, TREX1. So, this TREX is a also play an important role in sensing DNA molecule. Basically, this cytoplasmic DNA is efficiently degraded by 3 prime repair endonuclease, which is a TREX located in the cytosol. Okay, and oxidation of DNA, here you can see there is a oxidation of there is a non-oxidative stress and there will be oxidative stress or okay. So, here you can see that oxidation of DNA which occur in situation of oxidative stress caused by ultraviolet radiation or some kind of cell stress result to the oxidation of DNA molecule. So, this is quite interesting. There is a oxidation of DNA molecule. You have studied various redox reaction in various biochemical pathway like NADPH is involved in these kind of reaction. But here you can see the DNA also undergo the oxidation, the most common of which is 8 hydroxyguanosine. This oxidation product is 8 hydroxyguanosine, and this stabilizes the DNA against degradation by TRIX1, resulting in accumulation of DNA in the cytoplasm. So, this DNA will be accumulated in the cytoplasm, and eventually you have studied the C gas. Okay. So, this oxidized DNA is basically recognized by C gas, and once it is recognized by C gas, then you know this will make an intermediate, and this intermediate stimulate the sting, and then this sting will activate downstream signaling in order to produce the type 1 interferon and inflammatory cytokines. There is another sensor which plays a very important role in DNA sensing, but it is not inducing conventional cytokine, that is pro-inflammatory cytokine, or I can say that this induces a very potent pro-inflammatory cytokine, but it is not inducing the type 1 interferon. Okay. And this sensor is known as AIM2, and this makes a AIM2 inflammasome. Okay. So, here you can see that there is a AIM2 inflammasome, and this is basically consists of ASC molecule, as I have discussed in NLR when I was discussing NLR signaling pathway, and it is also associated with procaspase. Okay. And what happens once this complex forms, then there will be activation of procaspase into the caspase, and this caspase basically trigger the production, or this caspase basically cleave the inactive form of IL-1 family cytokine, which is present in pro form, and then this pro form will be converted into the active IL-1 family cytokine. So, here you can see that pro IL-1 beta is cleaved, and then there will be a production of IL-1 beta, and pro IL-18 is cleaved, and there will be a production of IL-18, which is active form, and this is secretory. Okay. In addition, this pathway also trigger the pyroptosis, and pyroptosis is basically triggered by gasdermin D, and this will trigger the pyroptosis. Okay. It's a kind of cell programmed cell death. So, AIM2 has a pyrene domain, and that is why this can interact with ASC. ASC has also pyrene domain and CAR domain. So, there will be a homotypic interaction, and the DNA is sensed by one HIN domain. There is a HIN domain in AIM2, and this is basically HIN domain and pyrene domain. There is some amino acid which is basically linking these two domains. So, I have already explained you aim to interact with ASC protein via pyrene domain, and double stranded DNA is binding with the HIN domain. Okay. The minimum length of DNA that induce the production of IL-1 family cytokine by inflammasome formation is basically 80 base pair. If there is a shorter stretch of DNA, then it will not induce the IL-1 beta production. So, activation of this AIM2 inflammasome basically require the phosphorylation and linear ubiquitination of ASC. So, this is one additional point. Now, I will talk about the IFI-16, how IFI-16 basically senses cytoplasmic and nuclear DNA. Here you can see that this IFI-16 is basically localized in cytoplasm as well as in nucleus, and when DNA approaches to this sensor, it interact with a sting, and then you know the downstream pathway. Sting will activate TBK1, IKK complex, and there will be a production of type 1 interferon and inflammatory cytokines. So, IFI16 is another member of 5-HIN family protein, and this could detect the DNA, and minimum length of double stranded DNA for binding HIN domain. There are two HIN1 and HIN A and B domain is a 16 base pair. So, here you can see that previously you have seen that AIM2 can bind with approximately 80 base pair, and if there is a shorter stretch of DNA, then IFI16 can bind and induce the immune response. The acetylation of this IFI16 has been shown to promote the IFI16 trafficking towards the cytosol, and C-gas protein is required to stabilize the binding of this IFI16 to DNA in the nucleus, and there is a crosstalk between both pathway. So, C-gas and IFI16 at some places they do have a crosstalk, okay. And IFI16 can also form a inflammasome. Probably you can understand, since this has a 5-HIN domain, so maybe it is involved in making the inflammasome. So, this is our study. Here we have discovered that this IFI16 is not only playing important role in sensing DNA molecule, it also senses RNA, and RNA is basically from influenza virus. So, here you can see that IFI16 can sense viral RNA. Okay, viral RNA is basically from influenza virus. They do not sense from other viruses, at least what we have tested, that is NDV, Newcastle disease virus. They do not sense NDV RNA, and basically this upon sensing they trigger the pyroptosis, as you can see in this slide. Okay, this pyroptosis, besides the production of inflammatory cytokine and type 1 interferon, they also induce the pyroptosis. So, now I will talk about a kind of, I will summarize various DNA sensor. Okay, and here you can see that about more than 40 to 80 base pair DNA, double stranded DNA can be sensed by C-gas, AIM2, okay, in human, and in mice it is sensed by the C-gas. Okay. Another is that there is a G-rich Y-form DNA, base paired DNA is more than 12 base pair, okay, and this unpaired G is adjacent in single stranded DNA, okay, and this is sensed by C-gas. There is a CpG motif DNA. You know that this CpG motif DNA is sensed by TLR9. AT rich DNA or AT rich which is present in DNA, that makes a AT or AU rich RNA, and this is basically sensed by the RNA polymerase 3, and basically RNA polymerase makes this AU rich stretch of RNA, and this stretch of RNA is sensed by the RIG-I, and in that way this RNA polymerase 3 indirectly sense the DNA molecule. So, more than 400 base pair can be also sensed, which is basically a long DNA RNA hybrid. It is in the case of HIV infection. So, this is also sensed by the C-gas, okay, and there is a short DNA RNA hybrid, and this short DNA RNA hybrid can be sensed by the TLR9. Okay. So, with this I will stop here, and in next session I will discuss about DNA sensor and disease and therapeutic, and with this we will complete the pattern recognition receptor, and we will also complete the 6th week of this course. Thank you.